Hi, my name is Erick. I'm a postdoc working at the Beckman Institute with Professor Aron Barbey. Today I'm going to talk to you about a large-scale, comprehensive, multidisciplinary training project called InSight. InSight is a multimodal intervention designed to improve cognition and intelligence. So it's a pretty big collaborative effort spanning many labs here at Illinois and other universities. So InSight combines 3 intervention modalities, each with known benefits to cognition. The interventions include game-based cognitive training, some custom software that we've been using across some labs at Beckman, direct current brain stimulation, and high-intensity fitness training intervention. So our baseline metrics span fitness, multiple cognitive domains, and multiple MRI brain measures. And the interventions have never been combined with such extensive baseline metrics before. So we have 300 participants in interventions lasting 16 weeks. And I thought what I'd do is use the remaining 4 minutes to try to talk about as much of this as possible. That's actually not true. I'm only going to talk about 2 small things. So I'm going to talk about just fluid intelligence and MR spectroscopy. So InSight aims to improve cognition, memory, and learning through this multimodal comprehensive intervention. But we think to better understand any improvements we observe from these interventions, we really need to understand the baseline relationships among variables prior to our intervention. So I'll just focus on these 2 little sub pieces of the project. So what is fluid intelligence? It is a discrete component of general intelligence, and it's typically defined as somebody's capacity for logical reasoning and problem solving in novel situations. So how do we measure something like fluid intelligence? Well, in the spirit of InSight, the comprehensive nature of InSight, we administer 3 tests of fluid intelligence, and all the tasks share a common theme. Basically, a participant must identify in the task an abstract pattern and demonstrate knowledge of the pattern by completing it or ruling out mismatches. So I'll just quickly kind of brief you on what those tasks are. So in this example, participants must select a set of letters that does not match the common pattern among all the options. In the second task, participants identify and complete a pattern in a series of numbers. And in the final task, participants must identify a pattern in an array of stimuli and select the item that fills or completes the pattern. Okay, so why measure 3 times? You might wonder, if we're measuring the same thing each time, wouldn't one measurement just be sufficient? Well, multiple tasks, generally speaking, help to sort of reduce sensitivity to task-specific variability, but it also affords us a unique ability to interrogate subfacets of fluid intelligence. So each task might be tapping on a slightly different aspect of fluid intelligence or adaptive reasoning and problem solving. So if all 3 tasks are measuring the same underlying cognitive construct, we'd expect in an exploratory factor analysis sort of thing, we'd expect to see only one common component emerge from the data. But in our pre-intervention data, so before collecting or before running participants through these interventions, we observe 2 components in this factor analysis. One that correlates most strongly with the BOMAT and the letter sets tasks, and one that correlates most strongly with the number series, the one where we complete the series of numbers. And together these components explain about 83% of the variance in the data. So it seems like there's a pretty clear dissociation between the 2 sort of subfacets of fluid intelligence. So is there anything about the brain that is predictive of these 2 subcomponents of fluid intelligence? So as I mentioned earlier, we are collecting MR spectroscopy data. And MR spectroscopy is an imaging technique that allows us to measure metabolites in the brain. And in particular, we targeted a volume of tissue in the middle posterior region of the brain. So with MR spectroscopy, you get a spectrum in this tissue, and each peak corresponds to the concentration of a particular metabolite. So for example, this large peak in the middle is this molecule called NAA. It's or N-acetyl aspartate. It's very, very common in the brain. I think the second most common molecule compound in the brain. And in general, it's sort of associated with neuronal health and neuronal functioning, and it seems to play a role in white matter, or the tissue that connects parts of the brain. So existing empirical evidence suggests that it is correlated, the concentration of this metabolite is correlated with cognition and intelligence, usually a positive relationship, so more NAA, higher performance in cognitive tasks. But existing studies tend to have relatively small sample sizes and often use general measures of intelligence. In the case of Insight, however, there is no other MRS cognition study of the scale. So in particular, we're interested in how NAA, this molecule we measure with MRS, relates to fluid intelligence. So in our data, we see a positive relationship between NAA and the first component of fluid intelligence, but not the second, controlling both for age and gender. So we see this as complementary to existing research, but it extends the literature by demonstrating an empirical dissociation between different competencies for fluid intelligence. So it may be the case that the first component is driven more by verbal reasoning, and the second component is more numerical reasoning, so maybe there's that nuanced difference there. So NAA seems selective to one particular component of fluid intelligence, and it's worth noting, I think we also see a gender difference in the second component, but not the first. So we find this apparent dissociation between facets of fluid intelligence pretty interesting and plan to explore it in the intervention data. But more generally, this result, I think, provides some footing to help us better understand enhancements in cognition and the neurobiological targets for interventions.